Every human cell contains a specialized vesicle packed with concentrated chemical energy. Within this tiny sphere, the internal environment is highly acidic, creating a space where biological structures are rapidly dismantled. This is the lysosome. It carries 50 to 60 distinct types of enzymes called acid hydrolases, tools so effective at tearing apart proteins, lipids, and carbohydrates that the lysosome effectively functions as an internal weapon. Biologists first identified these distinct compartments in the 1950s. Christian de Duve noticed that certain enzymes remained latent in intact liver tissue, but became highly reactive the moment the cellular membranes were disrupted. The lysosome maintains this reactivity using V-type proton pumps. These mechanisms actively force hydrogen protons through the membrane and into the vesicle, dropping the internal pH to 4.5. At this level of acidity, the once dormant enzymes become functional. This creates a necessary conflict. If the vesicle ruptures, the acidic spill triggers cellular death. But without this containment, the cell would slowly be overwhelmed by the weight of its own metabolic waste. To prevent that waste from building up, the cell must safely manufacture and move these destructive enzymes without dissolving its own internal architecture. The protection begins on the assembly line. The enzymes are synthesized in the rough endoplasmic reticulum and then transported to the Golgi apparatus for specialized processing. Inside the Golgi, each enzyme receives a mannose-6-phosphate tag. This acts as a molecular zip code, marking these specific proteins for delivery to the lysosomal pathway. Receptors in the Golgi read this tag and bundle the enzymes into transport vesicles. This ensures the chemical cargo is sealed by a membrane boundary before it ever touches the general cytoplasm. This rigorous molecular tagging is the cell's primary defense against accidental self-digestion. Once formed, the lysosome transitions through a life cycle of maturation. It moves from a dormant state to an active reactor. The cycle begins at node 1, the primary lysosome. Fresh from the Golgi, this vesicle holds inactive enzymes, waiting for a target. That target is often an external threat. When an immune cell engulfs an invading bacterium, the cell's membrane wraps around the pathogen, trapping it in an internal bubble. At node 2, the primary lysosome docks and fuses with that bubble. This process, known as heterophagy, allows the cell to digest materials taken in from the outside environment. As the compartments merge, the environment acidifies. The hydrolases wake up and rapidly break down the bacterium into its fundamental biological components. This stage serves as a frontline defense for the innate immune system, neutralizing biological threats before they can take hold of the cell. Beyond these external defenses, the lysosome manages an internal cleanup operation. Over time, every cell accumulates debris, damaged mitochondria, and misfolded proteins that threaten to clog the cellular machinery. Note 3 represents the autophagic vacuole. The cell wraps a membrane around failing parts for lysosomal salvage. Dissolved building blocks are pumped out for reuse. Finally, Node 4 identifies residual bodies. The lysosome isolates undigestible waste here in a dense vesicle, protecting the cell. This continuous loop of recycling and waste isolation is what allows cells to survive starvation and prevent toxic internal buildup. The impact of these microscopic cleanup crews extends to the macroscopic shape of the entire body. In the womb, a fetus begins with paddle-like hands. To create distinct fingers, lysosomes trigger the removal of the webbing between them, physically carving out the human form. We see this same structural engineering in our bones. Specialized cells called osteoclasts are dedicated to the constant resorption and remodeling of skeletal tissue. Unlike most cells, osteoclasts secrete their lysosomal enzymes into the space between the cell and the bone. They create a temporary, highly acidic pocket that dissolves the solid bone matrix to make room for new growth. From the curvature of your skeleton to the separation of your digits, your anatomical shape is the result of precisely targeted microscopic destruction. Because this system relies on dozens of coordinated moving parts, it is vulnerable to failure. A single genetic typo affecting just one of these 60 enzymes can disrupt the entire cellular environment. This results in lysosomal storage disorders like Tay-Sachs disease. When a cell cannot degrade a specific waste product, the lysosome swells uncontrollably, eventually leading to cellular rupture and fatal tissue damage. Cancer cells exploit the system differently. They often hijack the recycling process, using autophagy to scavenge nutrients and survive in the nutrient-poor environments within tumors. The lysosome serves as the cell's primary engine for maintenance and recycling, while simultaneously harboring the chemical tools for its total destruction. The same acidic compartment that drives human development and sustains life is also the cell's most significant point of failure.